Your blood may look like a uniform red fluid under the microscope, but molecularly speaking, it's more like a chemical symphony in constant motion, one with the power not only to deliver oxygen and ferry nutrients, but also to flip genetic switches that shape your future. Welcome to the fascinating frontier where blood meets epigenetics, the study of how environmental signals and biochemical inputs change the expression of your genes without rewriting the DNA itself. Epigenetics is like molecular punctuation on your genetic script. Your DNA is the same in every cell, but how it's read and acted upon depends on modifications like DNA methylation, adding a methyl group to DNA, often silencing genes, and histone modifications, changing how tightly DNA is wound around proteins, making genes more or less accessible. These are the dials and knobs that regulate gene expression, and, it turns out, many of them are adjusted by molecules that float around in your blood. Let's start with the influencers you already know, inflammatory molecules. Chronic low-grade inflammation, aka inflammaging, doesn't just damage tissues, it can epigenetically reprogram cells across the body. Circulating cytokines like interleukin-6, IL-6, and tumor necrosis factor alpha, TNFA, activate intracellular signaling cascades that reach deep into the nucleus, where they recruit enzymes that modify DNA and histones. For instance, IL-6 has been shown to increase DNA methylation at key gene loci that suppress inflammation, ironically making it harder for your body to turn off the very immune responses it triggered in the first place. That's a feedback loop with serious aging implications. Next, enter the world of blood-borne metabolites, tiny molecules produced by your cells, diet, and microbiome that circulate in plasma and quietly rewire your epigenome. One key player is S-adenosylmethionine, SAM, a methyl donor molecule that acts as the currency of DNA methylation. SAM levels in blood directly influence how effectively your cells can silence genes or stabilize the genome. Declines in SAM with age or oxidative stress can reduce methylation in the wrong places, potentially activating cancer-related genes or turning off genes needed for detoxification or repair. Then there's acetyl-CoA, a metabolite that serves as a substrate for histone acetylation. High acetyl-CoA levels typically lead to more open chromatin, meaning more genes are turned on. This is crucial for processes like regeneration and repair. But when acetyl-CoA levels are disrupted by high-fat diets, metabolic dysfunction, or mitochondrial damage, the body may become stuck in an epigenetic state that favors inflammation and tissue breakdown. Let's not forget NAD+, a molecule better known for its role in energy metabolism, but also a crucial cofactor for sirtuins, a family of proteins that regulate histone deacetylation. In plain terms, NAD+ fuels gene-silencing proteins that promote stress resilience, DNA repair, and metabolic efficiency. As NAD+ levels decline with age, these protective epigenetic programs also fade, leaving your genome more vulnerable to damage and dysregulation. Blood-borne NAD+ boosters like NMN or nicotinamide riboside are now being explored not just for longevity benefits, but for their ability to epigenetically reboot aging cells. Meanwhile, microbial metabolites produced in the gut and absorbed into the blood, like butyrate and indole-3 propionic acid, also have epigenetic sway. Butyrate, for example, is a histone deacetylase (HDAC) inhibitor. It prevents the removal of acetyl groups from histones, thereby keeping certain genes turned on. This is generally a good thing for genes involved in anti-inflammatory pathways, cellular differentiation, and barrier function. In other words, the fiber you ate yesterday could be influencing how your genes are being read today through chemicals now swimming in your blood. Even oxidative stress, often driven by elevated levels of reactive oxygen species, ROS, in the bloodstream, can push epigenetic changes. ROS not only damage DNA directly, but also modify the enzymes that regulate DNA methylation and histone structure. This can lead to epigenetic drift, a gradual, random loss of gene regulation over time, which is a hallmark of biological aging. Think of it as your genome's instruction manual slowly losing legibility, page by page, because of chemical smudges from a polluted bloodstream. The implications are profound. Your blood isn't just transporting these epigenetic modifiers, it's broadcasting environmental messages, metabolic status, and microbial activity to your genome. These circulating molecules essentially tell your DNA whether to gear up for repair, inflammation, growth, or shutdown. And yes, these changes can persist. Epigenetic memory can last weeks, months, or even across cell generations. That means a stressful season, a prolonged inflammatory condition, or even a bad diet binge could echo epigenetically through your tissues long after the initial trigger disappears. But the reverse is also true. Healthy behaviors, regular exercise, antioxidant-rich diets, proper sleep, can reshape your blood's epigenetic message and help stabilize gene expression patterns that favor longevity and disease resistance. Researchers are now exploring blood-based epigenetic clocks that estimate biological age by measuring methylation patterns across hundreds of CpG sites. These aren't just neat parlor tricks. They're being used to predict risk of disease, cognitive decline, even mortality. And with advances in precision medicine, we may soon be able to use circulating epigenetic markers to fine-tune interventions, recommending lifestyle changes, or even targeted compounds based on how your blood is literally rewriting your genetic behavior. In the end, your blood is more than a diagnostic tool or a delivery system. It's a biochemical editing suite. It doesn't change your DNA sequence, but it helps decide which parts of that DNA are whispered into action and which are permanently silenced. So, if you want to age well, don't just think about what's in your genome. Think about what's circulating around it, because that's where the real decisions are being made.